Theories of antibody formation and network, molecular immunology. Okay, there are many theories of immune response, direct template theory, indirect template theory, natural selection theory, and clonal selection theory. So we are not discussing about the first three. So we are concentrating on the clonal selection theory that is there in your syllabus. What is clonal selection theory? You have already come across this, and I have explained also in your earlier classes. So Burnett proposed the theory in 1957. The theory emphasizes the immunological specificity to the cellular level. In this theory, the cell are formed by somatic mutation. The cells that react to the self-antigens are eliminated and called as a forbidden clones. So their persistence in later life leads to the autoimmune process. So this theory was clonal selection theory was proposed by Burnett in 1957, and I'll explain it here. The postulates of theory: if B cells contact with a specific antigen, it divides rapidly to form a clone or identical cells. That process is known as a clonal selection. So different types of B cells have different receptor molecules. So there's different colors. So different types of B cells have different receptor molecules. When a pathogen or a germ locks onto a receptor, the type of B cell is selected. So this one. So this type of B cell is selected. So the selected B cell divides rapidly to make lots of copies of itself. The copies makes lots of antibodies against the pathogen, against that particular pathogen. So this is known as clonal selection. So some of the activated B cells becomes memory cells. Memory cell continues to produce small amounts of antibodies after infection have overcome. So antibody will be ready if the same infection occur again. The reaction becomes faster. So secondary immune system components. So these memory cells remain in circulation for a longer period of time. Next theories to explain the antibody diversity. There are three theories to explain the antibody diversity: germline model, somatic variation model, and the two-gene model. So first, I will explain the germline theory. So according to the germline theory of antibody diversity, the genetic information for each antibody is encoded in its entirety within the germline genome. That means the genetic information of the antibody diversity which is present in the germline genome itself. So this means that the gene encoding the end chain or light chain that the animal could ever make must be present in every chain, every cell. So in the germline genome only, the information of the antibody synthesis is present. That is known as germline theory. Next, somatic variation model. So somatic variation model maintained that the genome contains a relatively small number of the immunoglobulin genes. Genome contains a relatively small number of the immunoglobulin genes from which a large number of antibody specificities are generated in the somatic cells by mutation or recombination. So according to somatic variation theory, the genome contains a small number of immunoglobulin genes from which a large number of antibody specificities are generated in the somatic cells by mutation or recombination. That is known as somatic variation theory or somatic recombination theory. In 1965, Dreyer and Bennett proposed that antibody heavy and light chains are each encoded in two separate segments in the germline genome, and that one of each B region and the C region encoding segments are brought together in the B cell DNA to form the complete antibody heavy and the light chain genes. Okay, so this is known as two-gene model. Dreyer Bennett. So they proposed antibody heavy and light chains are each encoded in two separate segments in the germline genome, and that one of the each B region and the C region encoding segments, they are brought together in a B cell DNA to form a complete antibody heavy and the light chain genes. This is also known as a two-gene model. Next, Tonegawa's experiments. In 1976, Tonegawa Hojumi, he found that the separate genes encode B and C regions of the immunoglobulins. That means B is a variable region and the constant regions. Both are encoded by separate genes of immunoglobulins, and that the genes are rearranged in the course of B cell differentiation. That means, so separate genes encode the variable region and the constant region. So during B cell differentiation, these genes are rearranged. So for this, Tonegawa, he awarded the Nobel Prize in 1987. So this was the experiment showed by Tonegawa. So according to this, So this is the germline gene, and this is the rearranged one. So the digestion of the germline and the rearranged will give the different restriction fragments. So this is what is given here. So the digestion of the rearranged DNA with the restriction enzyme RE is a pattern of the restriction fragments that differ from those obtained with the digestion of an unrearranged locus with the same restriction enzyme. So same restriction enzyme is used for the germline as well as for the rearranged, and you will end up with the separate or we can say the germline fragment differs from the rearranged fragment. That indicates that there is a rearrangement takes place during the B cell differentiation. So next, multi-gene organization of the immunoglobulin genes. This I will explain it in the next class.